Hello students, welcome you all to a new video of Sunanda's tutorial. In this video, we will discuss about electrophoresis, particularly polyacrylamide gel electrophoresis. So what is polyacrylamide gel electrophoresis? Instead of agarose gel, here polyacrylamide is used, and this technique is particularly mainly for proteins, specifically for proteins. So PP, you have to remember this like this, that polyacrylamide gel electrophoresis is for proteins. Next, it is a technique for separating proteins or nucleic acid, mostly proteins, and the charge using an electric field through a porous polyacrylamide gel, which acts as molecular sieve for high resolution analysis, identification, purification in molecular biology, forensic science and medicine biology. Then here polyacrylamide gel is used. Okay, next, what are the principles? So it is an analytical method mainly used to separate the components of a protein mixture, and particularly the charged molecules here will migrate in an electric field towards an electrode with positive sign. So positive charged molecules will migrate towards the negative end, and negatively charged molecules will migrate towards the positive end. So this is the principle of PAGE or polyacrylamide gel electrophoresis. The general electrophoresis technique cannot be used to determine the molecular weight of biological molecule, because the mobility of substance in the gel depends on charge and size. Okay. Next, what are the steps of polyacrylamide gel electrophoresis? So at first we have to denature that sample. So at first we have to denature the protein sample to make it, to give them an uniform negative charge and uniform shape, to make it linear, to convert it into linear shape. Then we have to add buffer, add glycerol for density and a tracking dye like bromophenol blue. Then we have to heat it. Boil the mixture briefly for approximately 5 minutes to fully denature the sample or protein sample. Then we have to prepare the gel for polyacrylamide gel electrophoresis. Here two types of gels are prepared: stacking gel and resolving gels. So stacking gel, which will form the top layer of the electrophoresis, and the main purpose of doing that stacking gel to concentrate all the loaded proteins into a narrow band at the top of the resolving gel, and resolving gel will form the bottom layer. Okay, and resolving, the main aim of doing the resolving gel is to separate the stacked proteins based on their molecular weight, and this resolving or separating gel will form the bottom layer. Okay, so in this way two types of gels are prepared. One is stacking gel and another one is resolving gel. Okay, next, then the electrophoresis process or running the gel. So at first we have to assemble different types of gels. Place the gel in the electrophoresis tank or buffer chamber, both stacking gel and resolving gel. Then add buffer, fill the inner and outer chamber with running buffer. Then load the prepared sample into wells created carefully. Then apply current. Connect the power supply and run until dye front nears the bottom of the gel. Okay. So you have to carry out the whole process until the dye front or the dye completely comes near the bottom of the gel plate. Next, visualization. We have to stain the gel by Comasi blue stain for few hours, then wash it with destaining solution up until the bands are clear. Then photograph or document the bands. Smaller molecules always travel faster and larger molecule moves slower. Okay. Then what are the applications? At first, the polyacrylamide gel electrophoresis is used for protein analysis, then nucleic acid analysis, but most commonly protein analysis. Then in the field of biotechnology and forensic sciences. Then what is STS polyacrylamide gel electrophoresis? So in STS page or STS polyacrylamide gel electrophoresis, STS means sodium dodecyl sulfate. It is a standard lab technique used for separating protein by size using an electric field through a porous polyacrylamide gel, and this technique uses SDS as detergent to unfold or denature proteins and coat them with negatively charged. So small proteins move faster through gel's meshwork or gel filter, creating distinct bands which are visible after staining. Next, the principle of STS page. So it separates protein primarily by their size, not by native charge or shape. By using the detergent sodium dodecyl sulfate, at first the proteins are denatured into linear chain with uniform negatively charged, which then migrate through porous polyacrylamide gel matrix towards the positive electrode, with smaller proteins moving faster and further than the larger one. So in case of STS phase, STS is a detergent used to denature the proteins, making it linear shape and then coating them with negative charge. Then steps: at first we have to prepare the sample. Proteins are mixed with STS and a reducing agent to break the disulfide bonds, denature them and linearize them. Then loading: samples are loaded into wells which are created in the gel. Then electrophoresis: power or electric current is applied, then proteins move through the gel from negative to positive end. Then separation: smaller proteins move faster than the larger one and larger ones move slower, creating distinct bands. After separation, gels are stained using Comasi blue and silver stain to reveal the protein bands. Okay, so these are the steps of STS polyacrylamide gel electrophoresis. So with this, I am concluding my lecture video here on the topic polyacrylamide gel electrophoresis. I hope you liked it. If you liked it, please hit the like button, share with your other friends, and please subscribe to my channel Sunanda's Tutorial. Thank you.